Good morning, everybody, and thank you very much to the organisers for inviting me to speak about ESMART, which I'm presenting on behalf of the ESMART International Steering Committee. So what is ESMART? The European Proof of Concept Therapeutic Stratification Trial of Molecular Abnormalities in Relapsed or Refractory Tumours in Children. I think we'll just stick to ESMART. The international investigators of the study, the coordinating investigators, Birgit Gerger at the Institut Gustave Roussy in France, and those are the international investigators, and the writing committee is as you see it there. And very importantly, funding provided through Imagine for Margot, a parent-led French charity, and in the UK through CRCTU, ongoing at the moment. So as most people in this room know, in the context of cancer overall, childhood cancer is rare, although it doesn't feel like it to many of us here. It makes up just 1% of all cancers across all age groups, with a further 1% making up teenagers or young adult cancers. 1 in 500 children will be diagnosed with cancer in the UK before their 15th birthday, though, and that translates to your average GP seeing just 1 or maybe 2 cancer cases throughout their whole career. It may be rare, but it's still the most common cause of death in children under 15 in the Western world. Kathy's spoken already about the improved prognosis through research for pediatric malignancies, better management of emergencies, earlier recognition, improved supportive care, more effective regimens, and very importantly, international collaboration through randomized trials. But as many in this room also know, not everybody is cured, and the cost of cure as a result of late effects and toxicities of standard treatments can be very high indeed. So we do need new drugs, we need targeted agents, we need immunotherapy, but we need to do it in a coordinated way to benefit our children. So the overall goal is to improve survival for these poor prognosis childhood cancers by accelerating drug development. And this, as Darren has alluded to, involves a paradigm shift. Rather than taking any drug we can get from pharma, putting them into everybody and see which lucky few can benefit, having a more personalised and precision approach, profiling the tumours, looking to see what we can tell about are there prognostic markers, which children will do better or which will do worse with different drugs. Can we predict response and see whether the drugs hit the target, and thereby trying to target the therapy to patients on a much more individualized basis. So what is ESMART exactly? It's a European basket clinical trial of novel drugs. It's been run within the international European community through the ITCC, or Innovative Therapies for Childhood Cancer International Consortium, with different tumour groups in each country. And in the UK, we're fortunate that's through CCLG, Phase 1 centres, and the ECMC, Experimental Cancer Medicines Network. What the study involves is biopsy of tumour at the time of relapsed or refractory disease, profiling that tumour within the context of the national programme, different in each country, but in our country it will be through SMPEDS. Looking at those results through a molecular tumour board with the right people interpreting those results, and that includes bioinformaticians, pediatric oncologists, statisticians, and where possible, assigning treatment based on the molecular profile of the tumour, with the caveats that Darren has already mentioned. The study involves some arms of single agents and some of combinations, because we know we're very unlikely to make big inroads by just doing repeated phase 1 studies of single agents that aren't going to have a big impact in relapsed refractory diseases. And then very importantly, the study is adaptive, so that rather than having periods where there's no trial open, new arms can roll in and roll out of the study based on futility analyses, based on the evolving science, thereby cutting down a lot of the delays that sometimes come with the regulatory processes. So we've mentioned already the rationale is that these national profiling protocols, MAPI-ACTS, ITHO, INFORM, and in the UK, SMPEDS, will be determining these potentially actionable mutations. In adults, we know that stratified approaches are being employed to enrich clinical trials so that patients most likely to benefit are entered into these studies. So really, we want to employ that for children as well, trying to match the treatment to the patient's biology. However, again, as we've heard before, the diversity and heterogeneity of these detected molecular alterations makes doing trials really quite difficult with a rare population. And that's the real reason for needing this adaptive, innovative design where you can roll things in, roll things out, and have kind of a basket approach to these rather than single repeated trials. So showing where SMPs fits into the overall strategy, like the other international processes, MAPI Acts, Inform in Germany, ISA in the Netherlands, SMPs there. Clinical tumour boards feeding into the ESMART study, patients going on, consenting, registering, and then being assigned to treatment arms based on the tumour profiling. And I'll come back to that near the end. What's the real novelty of this study? So one really key thing is that we've had absolutely unprecedented engagement from really big pharma companies who have drugs of interest that some of us have been chasing for years and not been able to get access to. In the first wave of the ESMART, the first 7 arms, there are drugs from Novartis, from AstraZeneca, and from Bristol Myers Squibb, and there are discussions underway at the moment with Pfizer for some really key inhibitors, particularly in the ALK field, to include in the next wave that are coming out. So the principle is tissue biopsy, which is going to be mandated because of the fact that we know that tumours do evolve over time, so that you know that what you're looking at is what's actually in the tumour at the time of relapse, unless there's really an exceptional reason that you can't get a biopsy, and archival tumour may then be used in that situation rather than denying access, but that will be the minority. And then also there are combination studies. It's a combination of drugs with each other, or with chemotherapy arms that are relevant to a broad field of pediatric tumour diagnoses. So the hypothesis of the study is actually based on a relatively recent comparative review done by Xavier Paletti, who is the study statistician. We know from doing multiple early phase studies in children, looking back at both targeted agents and cytotoxics, historically we've always started at lowish doses and then gone up in successive cohorts. That adds time to the period taken to complete an early phase study. Looking back, we know that the toxicity profile, PK and doses actually are very close in children to in adults. And it's really rare to have an unusual toxicity in a child that's never been seen in an adult before. So what we want to do is try and cut down the number of dose levels needed by trying to see if you can get to the recommended phase 2 dose for a child much more quickly, seeing if the dose in adults, scaled down for body surface area, is actually the relevant dose for children, and only de-escalating if that's not the case, or if there's a really good reason to suggest that it would be significantly different. So we're trying to find the recommended phase 2 dose of a specific agent or relative combination of agents for children. And teenagers, of course, and then to explore early signs of activity in a molecularly enriched study population. So who will the patients be that are enrolled? It is for patients up to the age of 18, and there was a lot of discussion about this, but overall it's felt that over 18s can be enrolled in adult trials, and sometimes there are more trials of early agents that are available much more quickly. The exception is that for older teenage or young adult patients, 19, 20, 22, there's the fire alarm. If they have a particular pediatric type of tumour, such as a patient in their early 20s with a neuroblastoma or Wilms tumour, and Kathy has a few of them, it is felt completely appropriate to include them in these pediatric studies. They're standard phase 1-2 study inclusion criteria that include adequate organ function as usually measured, measurable diseases on scan, or for patients such as neuroblastoma or lymphoma, if they have only evaluable disease by MIBG or PET, that would be allowed as well. And then there are very careful performance status criteria, because you don't want to be putting children who are really too ill into these studies, because it doesn't do the child a favour and it doesn't do the assessment of a new drug a favour either. It is for children with refractory or recurrent malignancies, but it's across the board, solid tumours including brain, other solid tumours and lymphomas, and very importantly, leukemia patients are not excluded. So this model, where the drugs or the arms are of agents relevant to leukemia patients, they can be included if they meet the criteria, and there's a set of leukemia assessment criteria that have been developed by hematologists within the group. So assignment will be by the ESMART steering committee based on the molecular tumour profiling, depending on what's shown up in the tumour arms. And I'll show you a schematic of that, but it might be oncogenic drivers, gene mutations, translocations, amplifications, and sometimes protein expression. It is possible that patients may be eligible for more than one arm based on their biology, and in that case there's kind of a tree trunk approach to how you decide, is there a slot on the study, is the combination chemotherapy or targeted partner valid for the child's disease, and then pragmatics come into it as well. And of course it's possible, as we increasingly see, that with the targeted therapies patients may have benefit for some time and may go on to receive more than one arm sequentially. Importantly, although this is largely a targeted therapy type of study, there are broader arms, if you like, and there's one in it at the moment, where if there isn't a specific tumour target, you still want children to have access to new therapies because they've exhausted many other therapies. And that's where immunotherapy arms, such as an immune checkpoint inhibitor, has been included in the first wave of arms. And then where possible, each treatment arm will be enriched with patients who, based on their biology, are felt to be most likely to benefit. So, for example, patients with a PI3 kinase inhibitor will be put into the PI3 kinase mTOR arms, but there are other inclusion criteria for those arms as well. So the design is an international open-label prospective two-part study validating the adjusted adult recommended phase 2 dose for children and then doing efficacy assessments for each arm. Each phase 1 or 2, or each arm, if you like, is conducted separately as in separate trials within the overall main trial, but with common standards in terms of entry criteria, assessments, etc., except where the nature of the drug dictates that it should be something different. The study is being rolled out in probably 20 ITCC centres, and they are the ITCC phase 1 designated centres because they are, some of them, first-in-child studies. Within the UK, it's expected to be five UK centres, and they are within the regional referral networks who have been designated, and across the UK that gives a good geographical distribution. The numbers are broad, between 200 and 285, and that ranges because there are stopping rules depending on the number of patients deriving benefit in the different arms, and recruitment is expected to be over 3 years with an 18-month follow-up after the last inclusion, and an overall study duration of 54 months. So the study has just been launched in France. I must say that a big push for the study and the political pressure within Europe to open the study has come through the French government, the French MPs, and expected that the first arm will open in France in late July. And the UK, we're going through the process linked to the SMPEDS programme of getting that all teed up to open in 2017. And as I say, there'll be 7 arms at initial opening, 6 of which are combinations and 1 is a single agent, in this adaptive design which allows us to put in new arms depending on the emerging data. So the main objectives: validate the recommended phase 2 dose of the single agent or combination, to look at the pharmacokinetic principles, to look at tolerability, toxicity, and then moving on to the phase 2 bit to look at the preliminary activity, i.e. tumour response, and where possible is that linked to the molecular profile of the tumour. There are some secondary objectives: to characterise the side effects or toxicity in more detail, including in a longer term, to characterise single agent and multiple dose PK. So the PK is the levels of the drug in the blood and how that relates to response. And then, of course, does it make a difference to the patients? Progression-free survival, time to the disease coming back again or progressing again, and how many patients can be long responders on these drugs, longer than 6 months. And then again, whether more of the enriched populations are responders. And then there are some biomarker exploratory objectives as well. Does the drug hit the target? And then looking at things like circulating tumour DNA, etc., as Darren alluded to earlier. So these are the initial arms in ESMART, arms A to G. So there's 2 arms with CDK4/6 inhibitors in combination either with TOTEM, which is topotecan temozolomide, or with everolimus, which is an mTOR inhibitor. And then you can see there's a WEE1 inhibitor with carboplatin. a PARP inhibitor, which is a class of drug we've been trying to get for pediatric studies for a really long time, in combination with irinotecan. And then one of the very best AstraZeneca TORC1/2 inhibitors. There is a single agent arm because it really is a first-in-child study with no pediatric data whatsoever, but there is also a combination arm in combination with TOTEM. And then the immunotherapy PD-L1 inhibitor with cyclophosphamide, with or without radiotherapy, where appropriate. And the chemotherapy arms have been picked to be relevant to the broadest number of different tumor diagnoses, so a reasonable amount of thought and planning has gone into that. So that's the schematic of the study overall. You're not meant to see all the different bits of it, but clearly you can see in groups if you have if your tumor, these are some of the molecular abnormalities that may show up on the various types of profiling. They're not exhaustive lists, but then looking at if you're in this neck of the woods, you may be eligible for one of these arms. If you have DNA damage kind of suggestion in your profiling, you may be eligible for one of these. Arms E and F are the PI3 kinase, mTOR, really broad applicability arms, and then there's the immunotherapy arm, which is less targeted but provides access to patients. And just by way of one example is what the kind of dosing looks at. We've set all of them up. This is the PI3 kinase mTOR arm. So most arms will start at a dose level 1 with a de-escalation built in case the side effects are more than we'd be hoping for. But if it's extremely well tolerated, then it's built in to escalate within cohorts to try and get the best combination, the best bang for your buck, if you like, the least side effects with the highest chance of benefit. So just coming back to where it fits into the overall national and international process, we've mentioned the MAPIACs, the other country arms before. It's important to say that although our SMPs will feed into ESMART, ESMART is one of the options into which it will feed. And Darren's very involved in leading the setup of a molecular tumour board nationally where any of these things, you do look at the profiling, there may be ongoing phase 1-2 studies, either academic sponsored or pharma sponsored that we take part in already. There may be registered or authorized agents available for that. Some companies are setting up similar profiling, and Roche Genentech has their Matrix trial, for which we have 2 studies already open in our phase 1 centers, the atezolizumab and the cobimetinib study coming shortly. And then if there's no actionable targets, looking at non-targeted arms of ESMART or other non-targeted phase 1-2. So really, ESMART is a very good option, but there are other options that will fit in within the UK as well. So I just want to acknowledge the international ESMART team, particularly Gilles Vassal, who's pushed this forward, and Birgit Gerger, who's the international CI. And then at a local level, particular thanks to the CRCTU team, Pam Kearns, Nicola Fenwick and Eleanor Brogden working on this with me, and the SMPs team, Darren's team, pushing forward the clinical molecular tumour profiling. And to that I should add Professor Andy Hall in the CCLG Tumour Bank. Thank you very much.